Welcome viewers, welcome to the second lecture on prokaryotic gene regulation. In our first lecture on prokaryotic gene regulation, we have discussed the various steps wherein gene can be regulated. And we have realized that it is supposed to be the prokaryotic transcription initiation step wherein maximum regulation can be observed. In our first lecture, we discussed about the involvement of sigma factors. Today, we are going to start about the role of various activators in regulating prokaryotic gene expression. Most of the time, the activators that are working, they are just executing their action merely by getting recruited onto their DNA binding sites. But herein, we will discuss about certain specialized activators like NTRC and MERT, which are involved in regulating gene expression by allosteric mechanisms. So let's start up with NTRC. What is the job of NTRC? That NTRC is involved in regulating the expression of GLNA gene, which is involved in expression of genes to combat the unavailability of nitrogen sources. So here we will see that NTRB is another component. What it does when it visualizes that the system is getting depleted of ammonia or some nitrogen sources, then it undergoes phosphorylation. Once it gets phosphorylated, then it carries on phosphorylation of NTRC. And phosphorylated NTRC is the true activator. Once it gets phosphorylated, it gains its ability to bind to activator binding site, which is almost 150 base pairs away from the promoter region. Once it binds to its activator binding region, what it does that it alters the conformation of RNA polymerase, which was already bound onto the promoter region. But this RNA polymerase, which was previously bound to the promoter region, was locked in a closed promoter complex. And as we all know that to initiate transcription, the closed promoter complex should undergo conversion to an open promoter complex. And this is the point wherein NTRC executes its action. Binding to the activator, it carries on the conversion of this closed promoter complex to the open promoter complex. This is the first example. You can see that an activator is activating transcription by allosteric modification of RNA polymerase. Now let's have a second example wherein we will see that MERT gene, which is involved in breakdown of mercury. This operon is regulated by an activator called MER-R. Herein, we will see that MER-R has got binding affinity towards the promoter region -35 and -10 region of the promoter that carries on the transcription of MERT gene.
Ma'am, if MER-R binds between -35 and -10 sequence of MERT, then how it facilitates the binding of RNA polymerase?
Actually, the promoter region of this MERT gene has got certain exceptional features as it is not found in other sigma 70 requiring promoter sequences. First difference is that the number of nucleotides between the -10 and -35 sequences should be 15 to 17 base pair if it is a sigma 70 dependent promoter. But here we will see that the difference or the distance between -35 and -10 is almost 19 base pairs. And this unusual feature leads to generation of -10 and -35 sequences. They are facing two opposite ends of the helix. And moreover, this MER-R binds to a region between -10 and -35, but the phase of its binding to the helix is opposite the phase of binding of RNA polymerase. So both of them can bind to the DNA facing and binding to the opposite faces of the DNA. And what actually happens that MER-R, when binds to the region between -10 and -35, it locks the RNA polymerase in such a conformation that the -10 and -35 sequences remain in the opposite faces. Only when that Hg+2 comes into the system and it binds to the MER-R, then the interaction between Hg+2 and MER-R leads to a conformational change in this MER-R Hg+2 complex. And what it does, this alteration in conformation of MER-R leads to twisting of the DNA or bending of the DNA, and as a result of which the -10 and the -35 sequence, which were on the opposite faces of the DNA, they are brought onto the same face because the MER-R complexed with Hg+2 is twisting the DNA. So this is another example wherein we can see that an activator can activate gene expression just by altering the conformations. So in our next part, we will talk about gal operon. In gal operon, here we will see that this operon has got two regulatory genes, just like lac operon, where we have got repressors to repress the synthesis and positive activators like cyclic AMP CRP complex. But the difference between lac operon and gal operon is the binding site of the repressor is a separate place and the binding site of RNA polymerase is a separate place. But still we see that once this repressor is bound onto the operator region, it prevents the binding of RNA polymerase onto the promoter region, though promoter and operator don't have any overlaps. In our next part, we will talk about arabinose operon. Ara operon is a separate variety of operon because herein we will see that a single molecule called Ara-C can alone act as a repressor of the operon, and when it gets complexed with arabinose, it can act as an activator of the operon. If I look into the arabinose operon, it comprises of certain structural genes, namely Ara-B, Ara-A and Ara-D. They are being synthesized from a promoter called PBAD. We also have got another promoter called Ara-P, which carries on the synthesis of Ara-C. So in all, there are two different promoters which are carrying on synthesis of the structural genes, or which are in all involved in regulating the arabinose operon. To start up, let's have a look that Ara-C has got binding affinity towards 4 different operator sites. These operator sites are Ara-I1, Ara-I2, Ara-O1 and Ara-O2.
Ma'am, then which operator bind in which promoters? There are 2 different promoters.
Yeah.
Then which operators for which promoters?
So if we can precisely see that the promoter which is involved in synthesis of structural genes, we have got operator site Ara-I, which is divided into 2 parts, Ara-I1 and I2. This operator is responsible for regulating synthesis from promoter PBAD, and the promoter which is responsible for synthesis of Ara-C is regulated by an operator called Ara-O1. Right. So there are 2 conditions. Condition number 1, situation when there is no arabinose. If there is no arabinose in the system, then it being a catabolic operon, we really don't need the expression of enzymes involved in breakdown of arabinose, as arabinose is not present in the system. It means we want repression of this operon. Exactly that is being executed by Ara-C. We will see that one molecule of Ara-C binds to Ara-I1 and another molecule of Ara-C will bind to Ara-O2. And both of them have got the tendency to form dimers. And actually, dimerization is the driving force towards the bending of the DNA. As Ara-I1 and Ara-O2 are at a far away distance, so to facilitate dimerization, the only option left is bending of the DNA. Then we will see that because of this bending, this RNA polymerase is unable to bind to PBAD promoter, and hence it switches off the synthesis of structural genes as it is desired. Now let us come to the situation when arabinose is added into the growth media. When arabinose is added into the growth media, then bacteria really needs to synthesize the genes which are involved in breakdown of arabinose. It means the structural genes B, A and D are to be transcribed and translated. So here we will see that as soon as arabinose comes into the system, it binds to ARA-C, and binding of arabinose to ARA-C leads to a conformational change in ARA-C. And as there is a change in conformation of ARA-C, it no longer retains its binding affinity towards ARA-O2 and it leaves ARA-O2. Instead, it binds to another operator region, ARA-I2. And arabinose ARA-C complex bound onto ARA-I1 and I2 in a way facilitates the binding of RNA polymerase on the PBAT promoter. Herein, along with this regulation, we again see the regulation exhibited by cyclic AMP CRP. As we have seen in case of lac operon, if the glucose level goes down, then the cyclic AMP level goes up and cyclic AMP CRP complex binds onto the cap site. And once they are bound onto the cap site, they together with two ARA-C arabinose complex bound onto ARA-I region, both of them join hands together to facilitate the movement of RNA polymerase from PBAT promoter. So herein we can very nicely see that two regulations are occurring simultaneously. Basically, cyclic AMP CRP are called global regulators. In E. coli, there are at least 100 such genes or genes which are regulated by cyclic AMP CRP complexes. Now we will take up the regulation in tryptophan operon. Tryptophan operon is a bit different from lac operon or arabinose operon. Reason behind it is a synthetic operon or anabolic operon, unlike lac and arabinose operon that were catabolic operons. Anabolic operon means the enzymes which are synthesized in tryptophan operon are actually involved in the synthesis of tryptophan. It means when the cell is depleted of tryptophan, then only the operon should be switched on and all the enzymes should be involved in synthesis of tryptophan. So it comprises of 5 structural genes: TRPE, TRPD, TRPC, TRPV and TRPA. And here what we will see that when the cells are growing in the presence of tryptophan, then the cell really doesn't needs to synthesize tryptophan. So what happens that there is a synthesis of a molecule called aporepressor. Aporepressor doesn't alone have got the capability to bind onto the operator sequence, but when it gets complexed with tryptophan, which is called as the co-repressor, it means aporepressor gets complexed with tryptophan, means aporepressor and co-repressor together makes a functional repressor. And this functional repressor then binds onto the operator region. It means when within the cell there is tryptophan, then we get a functional repressor molecule which is binding onto the operator region, and thus the synthesis of structural genes are switched off. But when the cells are really depleted of tryptophan, it means there is no tryptophan and only aporepressor is there, And as I explained earlier, that aporepressor being unable to bind to the operator region alone, it means operator region is vacant and RNA polymerase binds to the promoter region and carries on the synthesis of structural genes. So herein we can see that how tryptophan is regulating its own synthesis.
Ma'am, is the negative regulation is strong enough to control the tryptophan biosynthesis?
Yeah, rightly said. Unlike lac operon, here we see that the negative regulation itself is not enough to switch off the synthesis of tryptophan in presence of tryptophan. That's why the system has opted for another regulation that we call as attenuation. So this type of regulation is the point wherein we will see that gene regulation is occurring after transcription initiation. It means we are regulating gene expression after transcription has already initiated, leading to we are restricting the synthesis further by carrying on premature transcription termination. So in triboperon, you will see that between the structural genes and the promoter region, there lies sequences called leader sequences and attenuator sequences. What is the function of the leader sequence and an attenuator sequence? In the leader region, we will see that there are certain nucleotide sequences which have got internal repeat sequences. Means whenever the leader sequence undergoes transcription and we get corresponding mRNA from the leader region, it has got the tendency to form stem loop structures. So there are two possible ways of forming stem loop structures. One is between segment 1 and 2 and the second one is between 3 and 4. And herein we will see that the stem loop structure after 3 and 4 is followed by stretches of U in the mRNA. And as we all know from the concept of transcription termination that stem loop structures followed by uracil residues in the RNA is generating a row independent transcription termination signal, as a result of which it will switch off the transcription machinery once it carries on the transcription of this stem loop structures. Now let us think of the situation that what happens when there is very less amount of tryptophan in the system. If there is very less amount of tryptophan in the system, one point to be noted down is that in the segment 1 of stem loop structure that is formed in the leader region, we see that there are two consecutive sequences of tryptophan. And we all know that in prokaryotes, transcription and translation are coupled. So when the leader sequence is getting transcribed, it is immediately being followed by the ribosomes. And if in the cell there is less amount of tryptophan, this ribosome when carries on translation over the codons which demands tryptophan, it remains stuck there as the system is unable to provide tryptophan. So if ribosome remains stuck onto segment 1 of the leader region, then what will happen? That the RNA polymerase is moving at a faster rate, then the segment 1 is unable to form hydrogen bonds or stem loop structures with segment 2. Then what will happen? That instead of stem loop structure formation between 1 and 2, we see that there is stem loop structure formation between 2 and 3. And then we will see that the prokaryotic transcription termination signal or row independent transcription termination signal, which we were getting in terms of stem loop structures between 3 and 4 followed by uracil residues, is not acquired. And if that is not acquired, then the premature transcription termination that we were calling as attenuation is lifted up, and RNA polymerase doesn't falls off and it doesn't undergoes premature transcription termination. It means it will move further and carry on the synthesis of the structural genes. as it is needed, because the system is very much in crisis of tryptophan. So they have generated a system which is working simultaneously, or it is working along with the negative regulation for tryptophan biosynthesis. So this is another thing wherein we can see that premature transcription termination is being executed to prevent the synthesis of tryptophan if it is available within the cell. Some regulatory proteins, we will see now that they do not require a signaling molecule to bind to them and to activate them. We will see that they just undergo certain chemical modifications. As in case of oxy-R, what we will see that it has got free sulfhydryl groups, and when it undergoes oxidation in presence of peroxides or oxygen, the disulfide bonds are generated and the sulfhydryl groups are getting oxidized, and once these disulfide groups are generated, they regain the capability of binding to the DNA, followed by regulating gene expression. So herein we don't see that some signaling molecules are coming and they are carrying on some modification into the system. It is just the status of the cell. If the cell is getting converted from aerobic to anaerobic status, then what will happen? That they are getting reduced and they are again getting converted to sulfhydryl groups, and hence they are not in a position to bind to the DNA and regulate the gene expression. And similarly, we have got another example called FNR. It has got Fe4S4 molecule attached to it, and when it gets reduced, then only it regains the capability of dimerization and it gains the capability to bind to the DNA and regulate the gene expression. So these two examples give us a clue that every time the activators need not have to sense a signal in terms of some external compounds. They just undergo oxidation or reduction, thereby lead to change in conformation, or sometimes they gain DNA binding affinity, sometimes they lose the DNA binding affinity. Now we will discuss about two-component regulatory systems, wherein we will see that covalent addition of chemical groups is widely used to control the activity of both enzymes and DNA binding proteins. Phosphate is supposed to be the most common group, and apart from this, certain groups like methyl groups, acetyl group, AMP and ADP ribose moieties are also used. One large class of regulatory systems that use a phosphate group are two-component regulatory systems. The two-component regulatory systems comprises of two proteins that cooperate to regulate gene expression. The first component is a DNA binding regulatory protein that only binds DNA when it is phosphorylated. And the second is a transmembrane sensor kinase that senses the change in the environment and changes the shape. This causes the sensor kinase to phosphorylate itself utilizing ATP and then transfer the phosphate group to the DNA binding regulator. The example of such a two-component system is ARC-AB system that senses aerobic versus anaerobic conditions. Means cell should have the system to sense whether it is residing in an aerobic atmosphere or it is residing in an anaerobic atmosphere. Under anaerobic conditions, ARC-B or ARC-B phosphorylates itself and then it phosphorylates ARC-A. The ARC-A, when it gets phosphorylated, it represses the synthesis of almost 20 genes that are only required for aerobic metabolism. Thereby, it is saving energy. On the other hand, it activates half a dozen of genes which are needed for its existence in absence of oxygen or when oxygen level is low. The pathway of phosphate transfer in a two-component regulatory system actually involves 4 protein domains. These domains are actually highly conserved among different regulatory proteins and are mostly of two types. Those where the phosphate is attached to the histidine residues and those where the phosphate is attached to the aspartate residues. In a typical phosphorylase system of arc AB, as we have discussed, the phosphate passes from histidine to aspartate, and in this case arc AB system, the first 3 sites are on the arc B protein and the fourth one is on the arc A protein. Means first of all the phosphate is bound to the histidine, followed by its transfer to the aspartate, then it is again transferred to the next histidine, followed by its final transfer onto the aspartate residue which is present into arc A regulator. So in all, if we try to summarize what we have learned in this lecture, we discussed about the involvement of NTRC and MERT in activating the gene expression by regulating the gene expression by altering the conformation of RNA polymerase. Next, we discussed the arabinose operon and the regulations which are lying in arabinose operon. Then we discussed about tryptophan operon and the attenuation, which is an added regulation, that is premature transcription termination, which is a new phenomena which is also there in tryptophan operon. Then we discussed about 2-component regulatory systems and the involvement of a sensor kinase molecule and an effector molecule which has got the binding affinity towards DNA.